Morning to all. I am Dr. Mahaboobali, Sri Vijayavidhyala College of Arts and Science, Nallamani, Dharmapuri, Microbiology Department, Assistant Professor of Microbiology. It is my topic, which molecular biology topic. It was a very interesting topic for the students who are studying in microbiology, biotechnology, MSc microbiology, MSc biotechnology. That's why I am selecting this topic. About last 2 decades, I was working various places because of so many environmental conditions, and also the people who are approaching me. It is my privilege to give this topic, transposons. What is transposon? It is very simple example. It's called jumping gene. Transposon is nothing but jumping gene. Why the genes are jumping? So we are sometimes we are taking genes are very essential role or for photosynthesis. The central dogma of photosynthesis, first of all, DNA is replication takes place, then transcription occurs, the DNA have converted to RNA, then RNA is provide, mRNA is provide for protein. So it is called central dogma of photosynthesis in the molecular biology. Nowadays in the molecular biology, the transposon is an essential topic in which so many scientists have been taking a challenge how to occur the transposon, that means jumping gene from one place to another. So the very important phenomenon in which the jumping gene is useful in the RNA technology. So RNA technology is a recombinant technology. It's a very noted subject, environmental or genetic engineering subject. So we can recombinations of DNA from one organism to another to make new varieties. So such a way that the transposons are genes about this is the DNA. The genes here is called X. The gene is transformed into this here. Sometimes because of mutations. So mutations already you know that certain things affected by the physical and chemical and biological way. Because the mutations, the genes have been just jump into the other place, which is a very important role for variations of species. Variations of species. This is the just introduction about the transposons. It's nothing but jumping genes. It is occur in the plants and also the animals. Sometimes it makes some inheritance capacity, sex inheritance to produce new varieties of plants and animals. So what is the different types of transposons is a very interesting one. The transposon is nothing but one is non-viral transposon. And second is viral transposon. Third one is bacterial transposon. So the various types occur in the transposons. Number one is non-viral transposons, and the second one is viral transposon, and third one is bacterial transposons. So we have to classify based on the bacterial transposon is called otherwise called highest elements. Highest elements are example of bacterial transposon. Bacterial transposon is one of the examples of transposons. The example is present in the bacterial cells, bias elements are there. For example, other examples of Drosophila. Drosophila is Drosophila melanatus. Drosophila. Drosophila is nothing but fruit fly. The Drosophila fruit fly contain the IS elements. So the IS elements are bacterial transposon examples and the viral transposon, retroviral transposon. Retroviral transposon. Retrovirus transposon, so many virus having transposons, and it consists of so many examples. And the virus also having the transposons and the TCS elements, and so many elements are there. And the non-viral elements are classified two types. That means the Simons and elements. These are all the examples in which the transposons occur. So many types of transposons are there. For example, non-conservative, conservative, and also the mutation types of transposons, non-mutagenic transposons, and so many varieties are there. So the transposon is a natural phenomenon in which the genes have been jumping from one loci to one locale. It's called one loci to another loci. So it can occur by various phenomena. So, for example, even though sometimes we have to see the viral transposons, we have to take the example from retrovirus, in which the main mix of it is called a retrovirus. This is after some time it can break down by the transposons, the kind of this is called DNA. The DNA is just breakdown by the retrovirus, like the resistance enzymes we are using in the ordinary technology. Resistance enzymes we are using in ordinary technology for killing the genes and just paste another gene which were used by the ligase enzymes. So resistance enzyme is one of the leading enzymes in which using for the cutting of the desired gene area. Nowadays, the very advanced technologies also there. CRISPR-Cas9 technology. CRISPR-Cas9 technology. Nowadays, it is used by molecular biology and organic technology. So many research have been done in CRISPR-Cas9 technique. It is using for cutting and paste of the area in which what the DNA. It is a very important recent topic, the molecular biology. So my dear students, it is a quiz. I want to talk. I want to give the just a quiz regarding the transposons. Transposon is a very easy topic, so don't take it seriously. Just watching my series of videos, you can understand easily to write your future examination. Thank you very much. Thank you.